let's just get slowly started. What I wanted to, now we can get started. What I wanted to talk today about is autoencoders, and to see a bit how this works. I prepared some examples in Python, and but maybe before I go into this example, I quickly have another look here at what we've done last time, because I did this in the end, and maybe I went through it a bit quickly, and there's a few things I wanted to point out again. So you remember last time at the end of the last lecture, I quickly had a first glance at this notebook here, where I talked about how we use GPUs to do gradient descent. And we used just some random example function. I just took this function here, which I more or less made up to have a function which has a minimum, which sits somewhere here and which doesn't look entirely trivial. Let's make this a bit bigger. And so, I mean, you can see here where the minimum lies. Of course, this thing has a minimum at x0 is 0 and x1 is 1. And you can also see it here, 0 and 1. Somehow I think the minimum is here, so for whatever reason, this is the wrong way around. It's maybe because of a cosine which shifts in the minimum. In any case, we want to find this minimum down here. And as I told you last time, the traditional way is to calculate the gradient manually. So I take this in, calculate its gradient by taking the derivative with respect to both arguments. I write down the gradient in Python notation. What I've done here is when I wanted to see whether I correctly copied from here to here. So I just calculated the gradient at some random position 4 and 3. When I moved a tiny little step to the right and checked the difference, and I did the same here. I moved a tiny little step up and checked the difference. So I have here manually calculated the gradient with respect to x0 and x1, and here I calculated it using my formula. And as you can see with my formula and with my manual calculation, I get the same value. So at least at this point, I calculated it correctly. Strictly speaking, I should maybe try a few more places, but overall, it seems that I got the gradient right, and now we can try our gradient descent. And here is what we've done. I started at some randomly chosen position. I just took the same that we had before, 4 and 3 again, which is where 4 and 3, somewhere here. And now I always walked in the direction opposite to the gradient in order to get towards the minimum. So I calculate the gradient at my current position x, get dfx is the gradient, multiply that with a smaller number, and subtract that from the current position to get a new position, which is then the old position minus 0.1 times the gradient. And when I print here where my new position and the new function thing, and as you can see here, the function value gets smaller and smaller and smaller. It never goes below 0, but that's expected because, as you can see here, this is a function which cannot be negative. So here it has come reasonably close to the minimum, and as you can also here see, the steps get smaller and smaller. So here it's, in the end, we really make steps only around 10 to the minus 6 here, and here also steps around the 4th degree after the derivative. So we are really at nearly 0 here and more or less 1 here. And in that way, we came to the minimum of this function here within 20 steps. Obviously, an important part here is this scaling factor. If we make it so who can tell me what will happen if I make it too small, if I make it too big? What's the trade-off here? Yeah.
So in both cases, that one case we land on our minima in right amount of time.
Mm-hmm.
So we need more iterations. In the other case, maybe if we are in the minima and we have a big learning rate, we can drop out of the minima.
Exactly. If the learning rate is too small, we will arrive eventually in the minimum, but it might take ages. If the learning rate too big is, we risk overshooting the minima, but we make a step which brings us over the minimum and then back. Obviously, what I've done here is a very, yes?
Is there a way to find the learning rate, or is it trial and error?
For start, it's trial and error, but once you found a good start, then you can do adaptation of the learning rate. An obvious adaptation is the moment you notice that you take a step in the direction away from the gradient, you would expect that the function value gets slower. The moment you notice that function value didn't get slower but faster, then what must have happened is that you have a function like this. You were here, you made a step in this direction, but you overshot and ended up here. So whenever you see that the function value goes up, you have overshot, and obviously your learning rate is too big. So one of the most primitive or simplest algorithms to say whenever the function value, the new function value is larger than the old function value, half the learning rate from now on. And the other way around, occasionally try doubling the learning rate and see if it still goes down. But there's a whole, there is a whole research area in optimizer scheduling, which is all about how you can adapt how you can use your function values to adapt your learning rate while you do your optimization. There's also additional ideas which you can add to this. The first one is called momentum. Instead of simply always going in the direction of a gradient, you add a little bit of the previous gradient to the new gradient before going in that direction. That is a bit like you can imagine if you have if these are contour lines and this is the minimum and I start somewhere here, then here you can see this typical thing. I start here, when I might be tempted to make my steps faster and faster. Suddenly I overshoot here because I'm going too far. When I get slower again. And another thing which you often have is that you have these long troughs like here. You are here and you have to go all the way here in the middle. And it's a shallow, long trough. And what helps there is to imagine your minimizer is a little ball which rolls down there and it builds up momentum. So when it has been going in this direction before, in the next step, it will again first do a step in that direction before following the gradient. That can make things better, it can make things worse, depending on how this looks like. But many of these classical gradient descent methods have as one improvement that you can that you say the step should be alpha times the minus alpha times the gradient plus beta times the last step. And beta is some value like 0.1 when you always keep a little bit in this direction.
I wanted to ask about that, but came up elsewhere and I never got to ask a question about it. My understanding was that momentum is, the way it was presented was especially when you don't have access to the true gradient, but you have noisy estimates of the gradient and you're trying to...
Oh yes, yeah.
Is that the only use case? You also want it when you have access to the gradient?
This is an interesting question. I would have to look it up, but my gut feeling would be you're right, especially when your gradient is noisy, when the momentum helps a lot. That's why it's common in machine learning, because we will get a bit later today to a case where our gradient will get noisy, and where we can discuss about whether this is a good or a bad thing. But yes, this might, whenever the gradient is noisy, then the momentum will really help us. The other possibility that I said about speeding up along a trough, you might better achieve by making the gradient longer and shorter. Another thing that people sometimes experiment with is that they calculate a learning rate for each direction individually. But that sort of gives a strange skew to the whole thing, because in principle you should see this already from the gradient before. What I mean is you multiply, of course, the gradient vector by this learning rate, but you might multiply each component with a slightly different value, which of course makes your step no longer in the direction of the gradient, which on the first hand doesn't really help. There's something called the conjugate gradient method, which essentially works well if the whole thing is roughly parabolic, because then by doing it in a clever way, you can anticipate the effect of the curvature. But I don't want to go into that yet, also because I always get a knot in my head when I try to think about the conjugate gradient method. Yeah.
Does the optimizer function this automated learning rate?
The SciPy optimizer, I think it does. If we quickly have a look here at the SciPy optimizer function, what you can see here is it has different scalar function optimization. Here you see it has, this one here, we optimize has this Brent bounded golden, which are gradient-free methods, which actually don't have a gradient at all. So they rather try, wait, this is scalar, so if you only have a single argument. Here we have the multivariate one, and you have here this whole long list of things which you have. Neldermead is the one which I mentioned a while ago with making the simplex and always swapping. It's a gradient-free one. Powell, I've forgotten. Conjugate gradient is the thing that I've just mentioned, which tries to guess the curvature of the ellipsoid, and then does, in that case, something like, which follows this thing. This combines this with Newton, and this is also a conjugate gradient. All these ones, I'm not even sure, and for the others, Krylov Trust, I would have to look them up. But all these, some of them are gradient specific, some of it aren't. I used to know how BFGS works, but I've forgotten. And then here's a whole thing which is able to get out of local minima by coming out of it. But what we will do today is we will actually, once we switch over from NumPy and SciPy to Torch, then we have a optimization thing where you have where you use an optimization method called Adam. Optimizer. And that was basically a big goal. So over the last years, maybe when is Adam now? That was here. 2014 was the Adam method. So in the 2000s years, two methods came up which tried to do really clever stuff to get the gradient and the momentum and the learning rate and so right. The one was called Ada gradient, and the other I keep forgetting how it was called. And then came this paper which tried to unite these two ideas in one thing and called it Adam for adaptive gradient with momentum and something else. And by now everybody uses, many people always use this one. So, but that's of course, one way attempt here is to just give you an overview of these methods, but you can imagine that that is quite useful to have, to really put effort into trying to get this stuff good, because all machine learning is based on this gradient descent. Like Adam is another one of these crucial components of how modern large machine learning methods like also LLMs actually work. So these things do a lot. But coming back to this one, so here we do it simply, we just follow a gradient with a fixed learning rate. And even that for this simple function that already worked quite reasonably as you see. And then what we also looked at last time is that we said we don't want to calculate the gradient manually, because here it's still simple. I mean, calculating this gradient with paper and pencil is quickly done, but if your thing is long and complicated, this is very error prone and very cumbersome. And the great possibility is that with automatic differentiation methods, you can get this done automatically. And I already hinted last time on how this works, and maybe it makes sense to explain this in a bit more detail. So let's say we have a function, let's take this function which we have here, where we have x naught, and I always make squares around data points, and then as you can see here, this thing gets squared to get x naught squared, and then we have the other thing where we take x1 and the number 2, we multiply them, then we take the cosine of, so when we have 2x2, when we take the cosine of that to get cosine 2x2, and then we multiply these two to get this thing, and to that we then add another thing which takes also this here, minus 1. So this is the computational graph behind this. And as I said last time, this is a DAG, because everything here is with a directed graph, everything has a direction, there will be no cycles in this thing. And if we now want to calculate the gradient of this final result y here with respect to x naught or with respect to x1, what we would do according to the chain rule is we first calculate the derivative of y with respect to these two and add them up. And then we calculate the derivative of this with respect to this and with respect to that, and combine these two derivatives according to the product rule. And this thing, which gets multiplied according to the chain rule with that one. And this again, here, this thing here, then gets squared, so it takes its derivative with respect to its input by saying I have to take 2 times the input. And now you can imagine how you can sort of push the result from the bottom to the top. If in the end I want to calculate the gradient of this thing with respect to x1, I first tell the result node, this one here, I want to calculate gradients. Please push your gradients. When this thing says, okay, I'm a sum of 2 boxes, so I have to tell these 2 boxes, your gradient, that they both contribute to this thing. This thing then has to tell these 2 things. Maybe it's easiest if I write it. So let's say I write dy, so the total differential with respect to y is the differential of the left half, x naught squared times 2 plus cos x 2x1 plus the differential to the right one, x1 minus 1 squared. And the left differential I can again write up in, if I now want to get the left differential, I can say it's this plus this. And now I have to say this is multiplied here. Yes, here it's multiplied. So it's something like 2x naught plus the that thing plus x naught squared times. No, will I now manage to get it right? Okay. But you see the essential part, you always push the differential up, and in the end, what you will then get is that all these different factors which are multiplied here by this repeated application of the chain rule, they all move up here and they arrive here. And in the end, the derivative of y with respect to x naught will be the derivative of y with respect to this box times this derivative. And so if I multiply this derivative here, which is associated with the x squared, with the derivative with respect to that box, I get the full derivative with respect to x naught. And that box, in turn, is the derivative associated with this computation times the derivative associated with respect to that computation. So each stepping up means a product in the chain rule, and hence each of these things can calculate the derivative for its factor and give this thing to the other one and says, now calculate your derivative, multiply it with the thing that I've just given you and push it up there. And that thing gets all the products down here and says, I have to multiply to this derivative here and add it to this. And in the end, I end up here getting the derivative of y with respect to all these x naught. And the same way, x1 gets derivatives from two parts. It gets derivatives via that path and via that path. And it will add it up. So the rule has to be that while we do the calculation, all these intermediate values, they have to be stored. So normally when you do a computation with a computer, the intermediate values are immediately forgotten. But here the intermediate values have to stay within memory as an object which can still communicate. And then you can tell each of these things, go backward, and then this thing, which still has maintained the connections to its operands, it will tell its operands, here is your gradient, multiply your own gradient and push it upwards. And because each object here keeps connection to its operands, so to the things from which it was produced by addition or multiplication or squaring or whatsoever. And in that manner, you start at the very end, tell it, calculate your gradient, push upwards, and each one pushes upwards, multiplies in the process, and when it arrives here, it gets stored here. And when it's somewhere stored, and when a second one arrives coming from that path, then it gets added to what's already stored. And in the end, you can then take any different boxes and ask it, what have you stored? And it will immediately tell you the gradient with respect to that. I could also, if I had stored this in some intermediate variable, I could also ask, what is the gradient of this thing with respect to that? Then it would tell me everything, all the products up to here, but not the products here, because they come only later in the chain rule. Yeah.
It seems to be an interesting choice that the additive and multiplicative constants are entered into the tree as leaf nodes.
I've drawn it like this. Actually, while I was drawing, I thought myself, I wonder if it really does that, or if it keeps the constant as it is. But actually, I think it would probably keep the constants, because as we will later see, for efficiency reasons, all these boxes are not individual numbers, but they are whole matrices of numbers, whole arrays of numbers. And so it wants to be able to deal with a whole array of multiplicative constants. And they, of course, have to be somewhere in the memory. But there will be a difference between this box and that box. Because this box here, it can happen that we calculate the derivative with respect to this box, while we will never want to calculate the derivative with respect to a constant. So when we start the calculation, we better tell this thing, prepare yourself that you later ask to give a gradient, while we won't tell it to that one. So we make a distinction between operands, which may be required to sum up a gradient, and operands which may not be required to do that. And everything which we may end up asking for the derivative with respect to, we have to tell from the beginning that we need the gradient. And this is what we've done here in this code. You see, I start with this x, and as I've said, it's not a single x. It's not a single value, it's an array of two values. And it has here, and we tell it, we will later require to calculate a derivative with respect to this x, so prepare to have a gradient. And whenever I calculate a new value, using this as an input value, tell the new value that it should also prepare to take a gradient. And then you see what happens here. I calculate the function f of x, so it goes here, and it calculates this, and this will cause a lot of intermediate values. Here we get a square, here we get a cosine of a square, and so on. And all of these intermediate values will be calculated and then retained, so that they stand there ready to take up, to accumulate gradients. And then I tell y, okay, when I tell x, so these two values, in case you already have a gradient from last time, 0 it. So that we start with 0 again. As I told you, it sometimes happens that a value gets gradients from two paths, and then it will add them up. So I have to make sure that before I calculate any derivative, all the gradient accumulators are set to 0. Yeah?
So is there a reason why I call it? Does it,
I come to this in a moment. Yep. And now we go, and then, so I first tell all the x's, if you have a gradient, 0 them. And then I will tell y, now push your gradients backwards up the stack. And it's called backwards because it always goes against the direction of a DAG calculation. And afterwards, I can ask x, okay, what have you now accumulated in your gradient accumulator? And by this, I get my gradient of y with respect to x. And then I tell x to, when I tell x now subtract from x the grade 0.1 times the gradient. But this is now not a calculation which should be included in all this. So I tell it, while doing this calculation, don't keep gradients. Don't build up a DAG. Just do it without keeping the DAG. So normally, whenever you do any kind of calculation with these tensors, which involves at least one tensor that has this required grad set, a DAG will be built. But if you put it inside with no grad, then it will avoid making a DAG. And hence, whatever you do here will not contribute to any later gradient stuff. Yeah.
Would it work if you didn't set hash over that? Because can you still get a DAG? It seems to me x depends on g after this line, and similarly g depends on x. The magnitude of the gradient of x depends on x itself.
Actually, it probably would work because here I set the gradient to 0 again. And afterwards, and that will also destroy the link of, now I know what you're thinking. You say the new x will depend on the old x. But this is, I've written it here as an in-place operation. What's standing here is x equals x minus 0.1 times 3. And the x left of the equal sign and the x right of the equal sign, these are not the same x, obviously. This is the old x, and this is the new x. And in this case, it is now important that these two will be different things in memory, and they have different gradient accumulators. And this is why, basically, once I calculate this, and then I overwrite the x with a result, and the old x with its DAG will be gone. So this is this typical thing in programming. You have to be careful. Where is the identifier, the variable, which points to an object, and there could be two objects in memory which are kept separate, but they are referred by the same variable, only at different points in time.
I'd also like to ask about the zeroing out of the gradients. It's always been a step that confused me in machine learning. So you mentioned you can get it from different paths, and I guess practically what that would mean in terms of coding is at some point you call y dot backward, and then you have some other function of x, and you also call it backward on that. You want the gradients to accumulate. And I can't imagine any situation where I could imagine doing that.
Actually, you could. For example, imagine you have a complicated calculation where you want to write to where you have lots of data, and your final value y is the sum of some function applied to all this data. And you can apply it to each data object individually and then add them all up. And now let's say you make this as a loop. And in each loop you call backwards. that in order to accumulate the gradient for the step you've done in this loop, and then the next loop you say, okay, now just add it to the previous gradient. So you might have something like your function is the sum over lots of individual function calls which are all done in a loop, and so you want it to accumulate that things, because you would have exactly this problem here. Let's say you have something like 4I in some range, and then you say y equals some function of xi, and now you're actually interested in the derivative of y. But at the end, after all this is added up, so we are interested in the derivative of all the individual y's. And then one way to do it would, of course, be to add up all the y's and then calculate the derivative. And somehow y would keep build a giant DAG with all the individual steps, but then it would have to keep them all in memory. Or you say, okay, now just keep this in there and forget the rest. And exactly, that's how I understand it, which would be one advantage that it allows you. The other point is simply, so here it actually happens if you call backwards twice on the same variable. It just says, careful, you have called backwards again without zeroing in between. Is this what you want? So in a way, it's meant the way that you do this like that, or this zeroing. So, and here, so this is now the function as we've done it here.
I see this correctly, the y dot backward function, like something into the x dot grad.
Exactly. So y dot backwards will write something into the because there's two intermediate tensors, one containing this value and one containing that value. And y knows that it's the product of these two intermediate tensors. And even though these intermediate tensors don't have any variable which points to it, y keeps two pointers to them. So y can tell these two, please add the following to your gradient and push onwards. And then this thing, of course, will say, my two operands with this tensor and that tensor, I still have a pointer to them, so I can communicate with them until at the end you end up in the very innermost part, which is where our x.
With the code as written, can you access the derivative of y with respect to x naught squared, for example? And of course you could set z equals x naught squared and then z requires.
Yes, exactly. I would have to do that in order to be able to access this.
Otherwise, you can't.
Otherwise, maybe if you really go into the innards, you could try to walk the DAG upwards yourself. But that would probably be really cumbersome. Because for that, you would already need to know how the DAG looks like and then navigate it. So I'm sure you will find each tensor will probably, if you look closely, you will find within the tensor somewhere the actual pointers to the other ones, and then you can follow them. But this is one of these things which exists, but you're not supposed to use them. What they call a private field. It's only to be used by the tensor itself. Even for Python, doesn't stop you from accessing private information. But it will usually mess up your calculation. So on here, what you've seen here I've done it first manually, my gradient step, by telling it, I want to have here this, I want to subtract here 0.1 times the gradient. And now I take this very code that I have here and I write the same thing like this here. You see what I've changed here is I introduced this optimizer and I told this optimizer, you're now responsible for the elements of x. That's the thing that I want to find a minimizer in. And I want to make a stochastic gradient descent. I'll tell you later what the stochastic means. For now, it's a normal gradient descent here. And I give it a learning rate, and internally it does exactly the same. Optimizer.zeroGrad says, look at all the tensors that I gave you into your care and put all their gradients to 0. In this case, we have given it only one tensor, namely x, and it does that. when I call y dot backwards as before. And now, instead of explicitly calculating here the gradient step, I just tell the optimizer, do the step. And then the optimizer can, depending what it wants, either simply multiply the gradient with the learning rate, or maybe add a momentum to it, or maybe adjust the learning rate, or do whatever. And the nice thing about this is, as you've already seen before, there's a whole bunch of different optimizers in Torch. There's the classic old-fashioned stochastic gradient, the one with momentum, the ADA grad, the ADA max, the ADAM optimizer, all these different things which were invented in the last 20 years. And if you want to see, does it work better or worse if I just use a different optimizer? When you don't have to re-implement all your things, but just replace the optimizer here and the rest stays as it is. And with this, we now go through this, and then in the end, it does this. Here in that case, it does exactly the same as before, 2.8922. You see it ends up exactly at the same place, because the SGD thinks if you don't tell it anything else, it will simply do precisely what the code have done up here.
What is the grad function, grad underscore fn, that's shown with the tensor output of non-linked nodes? At least that's
where?
So, for example, the first line of output after that cell.
Here.
First line of output.
Ah, this here. Yeah. Oh, yes, this is because I said here print x.
So,
and x is this whole thing. It tells me in typical Python thing. Python usually has this habit, unless you tell it otherwise, whenever you have an object, it tells you the class of the object and then its parameters. And the class of a tensor object is tensor, and then comes here the thing written like a numpy array, and then its extra parameter, which is simply requires grad is true.
I actually want to ask, in the representation output f of x, the second component is grad underscore fn equals add backwards 0. What is this function?
It's a gradient function, which it means, this thing means I'm a leaf node. If a gradient comes, just add it here. And this thing here means I'm a computation node. If a gradient comes, push it back using the derivative rule for sums. Because it says here add backward, because that is the y tensor, which is a plus. If here this were not a plus but a star, then it would here say multiply backwards. So for each possible computational operation, it has a gradient function which knows what the adding rule knows you just add the gradients for two operands. The multiplying rules knows you have to multiply one gradient with the other constant and vice versa and add these. And this is basically tells you this thing uses the add backwards function, which is probably a function within the interiors of Torch, which codes how to calculate the derivative of a sum. And so it essentially identifies this node's operational node. At least I think so. That's now admittedly not something I know, but something that I'm guessing. So, and now the question, what's a tensor? Why is it called a tensor? There's essentially two meanings to the word tensors. For the physicist certainly will remember a tensor is what transforms like a tensor. And they will remember the frustration when you hear this for the first time of not understanding what it means. And hopefully also the aha effect when in your second or third year you finally get it. I'm not sure if you had this already. But here this is not that kind of tensor. So what I mean by that in physics, we use these terms to identify how it transforms, how it changes under a change of basis. But here, what we mean by tensor is something which can be contracted over. So you all know that if you have a two-dimensional array, like A, and say this one has two indices, I and J, and we have a second matrix B, then we might call the indices here J and K and sum over the inner index J. And in that case, we get the matrix multiplication. And in matrix multiplication, we usually always say we sum over the index we sum over is the last index of the left matrix and the first index of the right matrix, and we force these two to be the same. And that's what we call a contraction. But in principle, if you now had a three-dimensional array, for example, A, I, J, L, and B, M, N, P, and we want to somehow combine them, I could have the idea that I might say, okay, I want to identify the index J with the index P here. So I make this also a J and sum over it. And I might identify the index L with that, with the one in front here. So I have identified the index, the second index of A with the third index of B, and the third index of A with the first index of B. In doing this, I get a new tensor product, which now is indexed by the remaining ones. I, N, right? Yes. And this is called a tensor contraction. Tensor contraction simply means whenever you have a n-dimensional array, it could be 2, 3, 4-dimensional, whatever you want, and you have two of them, and you make this n-dimensional tensor and this m-dimensional tensor and produce a new tensor, which might be L-dimensional, and the new tensor will have the sum of the dimensions of these two minus all the dimensions which you contract over. And contracting over means running a sum over them and deciding that this sum index will be put here and here. And you can see this is a generalization of matrix multiplication. And in the parlance of machine learning, we call a multidimensional array, we call it a tensor, whenever we anticipate that we want to make such operations. Matrix multiplications or their generalization. And in the whole machine learning and neural network thing, we will see that basically everything is these tensor contractions, but we will often run into cases where the matrix multiplication is too clumsy, and it's much easier and more understandable if you spread out these matrices a bit. Yeah.
I didn't quite understand the tensor contraction. You were saying you choose an index, and then you loop through that index, and you...
Yeah, so let's say I have two three-dimensional tensors, A and B, which both have three indexes, so they are three-dimensional. And now I can define an operation where I say, I want to contract tensor A with B by identifying A's second index with B's third and A's third index with B's first. And so this is sort of the description of the operation. A matrix multiplication would be described in that case as I have two-dimensional tensors A and B, and I contract by identifying the second index of A with the first index of B. And contracting always means identifying indices which have been declared to be corresponding and then summing over the products with respect to when running a sum over the indices and sum via products. So you see here, each index here either appears replicated in the other tensor and is summed over, or it appears in the output. So I, J, and L is summed over, and I and N are not summed over, so they appear only in one of the two and appear here. And you can easily see that this way is, this double sum applied to these two three-dimensional tensors will give us a two-dimensional tensor, because each sum removes two indices. I think it's pretty, first of all, this is just an abstract definition, but there will be, once you have a couple of cases where you need this, it will become much clearer. maybe you guys know about convolutions in image processing? Exactly. So let's look at a convolution, because that's maybe the prototypical textbook example. You have your image, that's a big matrix M, Ij, where Mij is the pixel value in the ith row and the jth column. And you want to convolve this with a kernel, K, such that each, how do we do this now? And the convolution operation is when we have here a AK Mij times kernel I plus H J plus K. And that gives us when we convoluted M prime Ij. So here we have a two-dimensional tensor and a two-dimensional tensor. In this case, the indices aren't simply identified, but we have a relation how that this index goes with I plus K. But it's still that this index has to be somehow coupled with that index and this index with that index. If you now maybe have a three-dimensional index where you have here a third tensor L, which is the color channel, one for red, two for green, three for blue, then you might say, okay, now it's a three-dimensional times a two-dimensional tensor, and the third dimension is simply left untouched. But it's still useful if you now have a general function where you can say, I want to do a convolution where the first index of the left tensor gets convolved with the first index of the right tensor, and the second of the left with the second of the right, and the remaining indices stay where they are. And the whole PyTorch is built around giving you ways to do these tensor contractions, either in the convolutionary way, so with index shifting, or without convolution, just multiplying as in normal matrix multiplication, and doing this in a manner that you can then run gradients through it. And that's why we call it tensor instead of array, because we want to stress that they are optimized for this, which also means you might know that Google has now introduced this, has now manufactured these chips, the tensor processing units, or TPUs, which is their way to make a competition to NVIDIA's GPUs, by saying, well, NVIDIA has made their GPUs actually in order to accelerate graphics display, but now everybody doesn't want to use them for graphics, but for AI. In AI, basically, 99% of all calculations are such tensor contractions. So we should make our chip that is really specialized in doing these things. And this is why Google calls it now not a graphics, but a tensor processing unit. And by this they mean our chip is optimized to do lots of these tensor contractions in parallel. Yeah.
So did I get it right? The dimension or tensor contraction is the dimension of the two tensors minus the two times shared.
Exactly. And in Torch, we have used, now we will, afterwards we will always use matrix multiplication because that's the familiar one, but it's just a special case. In the general, there's the torch.tensor dot thing. And for the tensor dot, it's a function, it just takes two tensors. And then some arrangement of the two lists of indices, you give it 2, 3 corresponds to 3, 1. And then you would call something like tensor dot A, B, 2, 3 corresponds to 3, 1. And then it will calculate this thing. And by just giving here this list and this list of indices, you say what should be identified with what. And I think there's also a variant of this function which then does it in convolution style. So, okay, now what do we do with this? The thing that I wanted to show you now, what we do with it is the autoencoders, and we'll see in a moment how the whole thing works. And let me see, maybe I should quickly try to query 13247. So I'm now switching from my laptop to our server, which has a big GPU, one of these NVIDIA A40 things, which cost 2,000 euros. And so we will use that one here. And so what we have here is all the packages that I want to lead, and we'll see now while we go through it what they are. And the first thing I want to use our usual adult neural stem cell data set. And you remember when we worked with R, we always used the Server package, which has the Server object where everything was put together in one object. And in Python, they had a similar idea. So and they called it the undata object. And here, what undata is maybe best explained with this thing. So an undata object is an object which contains all these things here. And it works as follows. You have in the center, you have a matrix X, which is your actual data matrix. And the data matrix has data on different things which you have observed, which are called the observations, and they are the row. In our case, each cell is one observation. And for each cell, we measure a couple of different things, which are called the variables. In our case, these are the genes. But you see, they try to keep the terminology in general. The objects on which you measure are called observations, and the different quantities you measure about each object are called variables. And this time, in contrast to R, we use the statisticians and not the bioinformaticians convention. So the objects or observations are the rows, and the variables or features are the columns. And what we will typically have is that for each observation, we need a name for the observation. For example, the barcode of a cell. But typically, we have for each observation not only our barcode, but also a lot of other metadata, maybe from which batch it was, how well the quality control worked, and so on. So we have a whole table, and each row in the table corresponds to one row of a matrix. In the same way, we have a whole table where each column of a matrix is explained by one row in this table. So this table might have the columns for the gene ID, the gene name, the type of gene, whether it codes for a protein or not. So all the metadata, which is not really part of the measured data, but other stuff. And that goes in here. So we have here a table which explains the columns, and here a table which explains the rows. Sometimes the things which you want to explain, the elements of your columns, are not scalars, but are themselves vectors, so that you end up again with a matrix, but a matrix with a different shape. And this we call here the variable matrices and the observable matrices. And actually, I've forgotten what obsp and varp is, but doesn't matter. And here you can, and we will now see how we fill it. You can also see you can stack several matrices. For example, in our case, the counts and the fractions and the normalized counts and all the different stuff. So I do this here by telling this thing now that I want to build up an undata, and my big main data object should be the matrix. And the ops and var I also take from these two tables that I have with information about the cells and information about the genes. And then I also make this cross-reference saying that the main data matrix should also be given the name counts. And then in the end, I now get this undata thing, which has a big X object, which is again one of these sparse matrices with 18,000 rows and 20,000 columns. And the 20,000 columns are described by these, and the 18,000 rows are described by this barcode, which table, which tells us for each of the cells barcode and what cell type it is and what the other analysis happened. And we will later add more columns to this thing. And in the same way for the variables, for now we have only the G name and the ID. So, and now I want to do a standard analysis, and typically in Python there is a thing called scanpy, which is the same as Sera, but only for Python instead of for R. It's not named for a French paper, but it's simply named for single cell analysis with Python, or SCAN-Py. And they make a little Scampy as a logo. So, but I don't use Scampy now, because when I have to explain to you a new package, so I simply do it manually, all the stuff that Scampy would do automatically. So I do what we've always done. First, I calculate for each gene the total, so I calculate the row sums of the table. Each row is one cell, and if I add up the counts for one cell, I get the total count for this cell. And I add, for this, I now will add here to the cell table a new column total here, which contains these totals here. And I add a second layer, which is sort of like this green thing, but a second layer, which is now not the counts, but the fractions. It means the counts divided by the totals. An annoying little thing here is, if you've ever worked with pandas, you might already have noticed this. Whenever you want pandas makes a distinction between a table column and a vector. A table column is called a series, and it contains the vector together with an information of which rows it came from. And if you want to work with it, you have to say, I don't want the whole series, I only want the actual data column without the stuff around, so we have to put this to numpy. And another thing, in R, we always had this issue with that we had to transpose and transpose back. The NumPy people had another idea, namely something like this, means turn this vector into a matrix with as many rows as we had before and one column. So I do this, and this none always means at this position add a singlet dimension, so a dimension which only has a single entry. Like turn a vector into, if I write colon, comma, none, this turns a vector into a matrix with a single column. If I write none, comma, colon, this will turn a vector into a matrix with a single row. So that's R's way of making clear whether something's a column vector or a row vector. And the slash operator will use this to figure out whether it should divide everything, every column by this vector, or every row by this vector, depending on whether it's a row or a column vector. So that's actually done more nicely than in R, because if you get it wrong, it will immediately tell you that the dimensions don't match. The next thing we wanted to do is always calculate this variable genes, where for each gene we calculate whether it has more than Poisson variability, and for this we need to calculate the variance of the fraction matrix. For each row we have to, for each column we have to calculate the so for each gene that is, we have to calculate mean and variance, and unfortunately SciPy doesn't have a function for the variance of a sparse matrix, so I quickly wrote one here and put it here, and then I can use this here. So this function, you see I've written it, it gives you back the column means and the column variances, which are calculated here by this thing. And now I add to the variances matrix here these two things, and if I do this, then id.var, see, now has 1, 2 gene name and gene ID that we had before, and now I've added these two columns, which I've plotted for you here. So this is again variance over mean, and this is the mean. And as you can see here, I picked everything where the variance over mean ratio is above, I think, 3e3 or something like this. What did I say? Above 4 times 10 to the minus 4. And if so, I called it, made it a red. Here I've written whether it's red or black. So, yeah, I should have done is here I add the fifth column is HVG. And here I plot the whole thing. And these are now our highly variable genes. So from our 20,000 genes, I picked 4,000 genes which seem to have interesting information, and now I want to get rid of the others. So I say here, make me a new undata object where I contain only those variables which have a true here. And because it's an undata, what it will do is that it will remove all the genes which are not highly variable. It will remove it from the var table, from all the layers, and from all this stuff. And then I get here this new thing, which is the ndata object, only the highly variable genes. Then I calculate my log expression, as we've always done it, with counts for the gene divided by the total counts. So far, we always said times 10 to the 4 plus 1, like this. This time, I've written it plus 10 to the minus 4, which you see is essentially the same as here. It only means that the results of our old normalization and this normalization, they will be shifted by a constant of the logarithm of 10 to the minus 4. We will later see why this is more convenient to understand the autoencoder, because what this means is that k over s is actually the fraction, and the fractions are truncated to the below. They cannot be lower than 10 to the minus 4, but if a fraction is around 10 to the minus 3 or 10 to the minus 2, then you can understand this expression as actually simply the log of a fraction. And that will be later, because otherwise we'll get confused, or at least I got confused. So what I now did is I want to do a PCA here. And you remember, for a PCA, we take this log expression value, and before we run it, we standardize it. We subtract the mean, and then we divide by the standard deviation. So this is why here I calculated now for each gene the mean and variance after taking the log, which is not exactly the same as before. And I subtract here the mean, here, and I divide here by the square root of the variance. And now I have here the standardized log expression, LXPER, which is now the matrix of log normalized expression for all the highly variable genes, standardized such that every gene has mean 0 and standard deviation 1. And that's good to put it into PCA. The PCA function in Python, you get it from the sklearn package, which is the machine learning package that everybody uses. And it's a bit odd. You always have to set up your learning thing first by saying, what do I want? I want a PCA truncated 20 things. And then you have to say, and now apply this machine learning to our data. And then you can get in it the components, which are the loadings, and I can get the scores by now actually running the PCA transformation here on our data. So we have to specify the data twice. And now I get my scores and loading matrices. And again, the loadings matrix will have 10 values, 20 values per gene, and the scores matrix will have 20 values per cell. So it's exactly the kind of stuff which fits nicely into these matrix storages here, which store matrices which have where the number of rows corresponds to the number of genes or number of cells. So I put it here in the ops_m and var_m thing of my annotated data thing. And now with that, I can now tell this here and run UMAP on it, which also has a bit a strange way of how it's called. And see if it works here now. And you see now it runs UMAP. I store it as another matrix in ops_m. This is now a matrix with only 2 columns, but again, 1 row for each cell. And I plot this here. And now I get this thing here. And you see this is the data I'm always using as example. Here are somewhere the stem cells, and here's the cell cycle, and here's the neuroblasts. Or maybe the other way around. I keep forgetting. What I now want to do is I want to do something that we call denoising. And to explain what this is, I picked one random gene that's not a very biologically interesting gene. I have no clue what it does. I picked it as being a good example. And it's a good example because it shows us here what people call the salt and pepper pattern. Salt and pepper always means you have something white with a few black dots sprinkled in, like you have it here. You have here a light background and a few black dots sprinkled in. And on first side, if you see something like this, so what I plot here is simply the log expression of this gene cytochrome B alpha. And as you can see here in this area, the gene is sort of expressed, yet strongly expressed, yet nearly not at all expressed. And the question that we can now ask is, you see here there's a few green dots. Does this really mean that a few of these cells here do express the gene and most of them don't? Or that here, half of the cells express the gene and the other half don't? If we now were to go back to the count data, we would notice that all these cells here which express it, they have one count, and all the cells which don't express it have 0 counts. And so the more parsimonious explanation here would be to say all these genes, all these cells express the gene probably at the same level, but due to the fact that we only sequence about 5% of all the RNA transcripts, we sometimes see one gene, sometimes we don't. Imagine we capture 5% of all the transcripts, and there's a total of 10 transcripts in each cell. When, of course, if we capture every 20th transcript from a total of 10, then this means in half of the cells we will see 1, and in half of the cells we see none. And very occasionally we will see 2 or 3. So we might ask, is all this variation here, is this real biological difference between them, or is this just Poisson noise? And one possibility to do this is, of course, to take this whole blob here, decide, okay, we want to test the hypothesis that all these cells are actually the same and the salt and pepper pattern is only Poisson noise. So we calculate how much variance we would expect due to Poisson and compare with that. But this is a bit annoying because you see, in reality, we see there's a change. Here the gene is clearly strongly expressed, here it's clearly weaker expressed, and there seems to be some gradual change. So to go ahead, it probably looks like this here. The expression gradually increases from here to here. And this is probably a more proper showing of what's truly happening than this stuff here. So in a way, this here is a denoised example of that. This here shows Poisson noise, and here the Poisson noise has been magically removed. And the question is now, how have I done that? And you can already imagine if you have a lot of cells which are sort of similar, then it makes sense to assume that the cells, that we can maybe average over cells. If I have 20 cells which are all sort of similar, they have very similar PCA coordinates, and half of them express 1 count and the other half express no counts, and it makes sense to assign to all of them the equivalent of half a count. And one way to do that is to use our PCA. Because you remember what the PCA does in the end is, if I put this reconstruction here, let's write it on the board. There's 2 ways to understand the PCA. So far, we always understood the PCA as a projection. We project into a reduced subspace. And we assume that this projection is such that we retain most of the signal and lose most of the noise. And so if, for example, we have points like this, and we decide to project them all onto this line, when we get the following, let's say this point here is in reality, it's here, but it's spread out over there by noise. When if you project onto the line, then we lose that part of the noise, which is orthogonal to the thing, and we only keep the noise which is in the projection. If we project down from 4,000 genes to a 20-dimensional space, this dimension here is 3,980 and this dimension is 20. So we lose most of the noise and hence also most of the Poisson noise. And this is why using the projection might help. But the projection is with 20 values and we don't see our individual gene anymore. But what we can do is we can go back to the individual cell, to the individual gene. So if we imagine that we have our Y, the expression vector for cell I, which lives, let me write it like this, which lives in a space with, I don't know, 4,000 genes. And this one we project onto a Z, which lives in R20. And of course, we can try to undo the projection by rotating this 20-dimensional space back into the projection hyperplane. And then we again get a reconstructed Y, which I write Y hat, which is again 4,000 dimensional, but all the Y hat from all the different cells will lie in the same 20-dimensional hyperplane within this 4,000 dimensional space. And by going from here to here, we have reduced, we have lost hopefully mostly noise and only a little bit of a signal. And in order to do this, what I have to do is, you remember we have a loading, this core matrix, which is the matrix of all the Z, and we have a loading matrix which brings us from the Yi to the Z. So we can say Z equals R times Y, and this here is the loading matrix. And if I want to go back, then I make Yi hat equals R transpose Z, or equals R transpose R Yi. So this is the projection matrix, RTR, which goes to the 20-dimensional and rolls back so that we then end up on our projection. And we can now ask how good is the reconstruction. So we ask, what is the reconstruction error? What is Yi hat minus Yi squared added over all the I. And you remember the PCA tries to make this thing as small as possible. Yeah? No, wait. R is a 20 by 4,000 matrix. RT is a 4,000 by 20, so the 20 is in the middle. And this is a 4,000 by 4,000 matrix. That's exactly what makes it a projection matrix. That's the hallmark of a projection matrix, that it's square, but its rank is much smaller than the original, than the outer dimension. And so this is what I've done here. In order to do this, I have taken here my scores, the Z's, multiplied it with the transposed R matrix here. Don't get irritated that on the board the R is to the left of a Z and here the R is to the right of a Z. These stupid things always, it's always the other way transposed in the code than on the board. I'm trying to not have this, but it doesn't happen. And if I do this, then I get here my reconstructed R, that's this here. But you remember the PCA tries to approximate our standardized genes because we standardized them before we put them into the PCA. So if we get now the reconstructed out of the PCA, we have to unstandardize. So we again have to multiply with the variance and add the mean to undo what we've done here before, where we have subtracted the variance, where we have subtracted the mean and divided by the variance. And what have I done here? What's wrong here, if I now look at that, I'm an idiot. There's a square root missing. So this is now dangerous. I'm now changing something here, which was wrong. And let's see this, if now everything breaks down. NumPy dot square root. And with this, I can now make my improved scatter plot. Oh, looks slightly different. But you see now how this flipped. Yeah, this is the old one, this is the new one. You might remember from the exercise, these UMaps have a habit of sometimes flipping. So here, this is now the noisy one, and this is the denoised one. And that worked reasonably well to get it smoothed. But there's one catch. The PCA is a linear operation. And it's in a way not really reasonable that a linear operation is the best way to denoise this. So can we do better with a nonlinear operation? And so to do this, now this also looks much better. Here first to see what's actually happening, what I've done here is I put here the original data on the x-axis and the reconstructed data on the y-axis, still only for this one gene CYBA. And each point is a cell. And as you can see here, the original value grows from the left to the right, and the reconstruction also grows. And there's here this gray line, which shows you perfect reconstruction. And it seems that this has a tendency to be too far right, but here's all the zeros which sit on top. And I used a color scale to show us these are the cells which had 0 counts, these are the cells with 1 count. Now this is 0 count, 1 count, 2 counts, 3 counts, and more counts. And then you can see, use this to understand a bit how the discreteness of the counts sort of makes it a bit hard to really say that whether this is a good reconstruction or not. So what we now do, and it's must be a point why we talked so much about optimization before. So we now want to redo, and that's the first preliminary step before we go to the actual nonlinear autoencoder. We remind ourselves how the PCA is also an optimization problem, because after all, we want to minimize this thing. And we can minimize this thing in 2 ways. We can use our overall logic where we said, if we use the eigenvalues to make the loading matrix, if we assemble the loading matrix out of the eigenvectors, then this will automatically be minimized, or we can simply minimize it brute force. So here is this calculation that I've done here, that I've now used here to calculate my minimization. I take here the scores matrix, so the Z, this is VR, and I compare it with the original. And I square all the differences here in order to get, and then calculate, well, instead of a sum, I took the average. And we get 0.62. I can also write this in a bit the same calculation with the same result. I can write in a slightly more suggestive way by saying my original data, it's, well, my standardized log expression data. These I turn into what I will call the latent coordinates. That's the coordinates on our 20-dimensional hyperspace here. And then I reconstruct from the original from the latent by turning the latent back in this. And then I compare the reconstructed with the original. And the reason why I've written it that, because now you can say there's 2 operations. This here we call the encoding, which turns original into latent. And this here we call the decoding, which turns latent into reconstructed. So this means with PCA coordinates, the latent coordinates, are supposed to keep as much information as possible about the cells, such that we can reconstruct it as well as possible. So we now consider the PCA as a compression algorithm, which tries to compress this 40-dimensional data into 20 dimensions and then decompress it again. And here I now write this as an optimization problem. I say, this is my encoder matrix, and this is the decoder matrix. And I say, I take my data. The data, as you see, is again all the standardized log normalized expression. and I encode the data by multiplying it with my encoder matrix, and I decode them by multiplying them with a decoder matrix. And so I get from here to reconstruct it and back. And for this, the encoder matrix should be, of course, be a 20 by 4000 matrix, and the decoder matrix a 4000 by 20 matrix. You see, I transpose the decoder matrix. And in that way, and I initialize these two matrices with random numbers, just to start somewhere. In the PCA, these two matrices are the same, or rather they're transposed by each other. But just because it turns out that this improves convergence, I make two different matrices. In principle, they should converge to each other's transpose. And now, I put this in here, I calculate the loss, and now I want to calculate and I'd use my optimizer. And now here you see the magic of PyTorch. I can just say, calculate the gradient of this loss respect to all the to each element of each of these two matrices. And it will automatically have built up a DAG which captures this whole calculation, including all these matrix multiplications, and we get a correct gradient. And by this, I can run my optimizer. If I do that, it does this here, and as you see, the loss, so the reconstruction error, this thing here gets smaller and smaller every time. Questions?
So, update our encoder and our decoder.
Yes, and this is what happens here in the optimizer.step. We tell the optimizer here, you are responsible for optimizing the values in these two matrices. So whenever it does optimizer step, it will change these two values, the values in these two matrices, so that next time the loss will be smaller. Mm-hmm.
And is it important to choose good encoder and decoder start values?
In principle, it might be helpful. In practice, it's hard to do that. And one way is to try to find something good so that we don't get caught into local minima. But as you see here, I made my life simple and just use random numbers, and they were completely off in the beginning. And we're still far off. We're at 2,722. We want to go to 0.62.
What is the use of this transposition and then left multiplication? I was expecting no transpositions and always left.
Yeah, I think the problem is that our data, usually when we think about, when I write things like this, I write the y as column vectors. But you remember we just said that we use the statistician's convention that each observable is a row vector. And hence, everything which is a column vector here is a row vector here, and so everything either has to be transposed or multiplied from the other side. To be honest, when I prepared this code, I simply kept trying it out until I got it right. That's also the other advantage of this tensor multiplication team that I mentioned earlier, where you simply completely forget about this transposition. You simply look at your thing and say, okay, this index is the gene, this index is the PC, in the other thing, that index is the PC, so let's contract them. And you don't care anymore whether it's transposed or not. You just care, these two indices describe both genes, so I want to contract over them. So, but as you can see here, it's sort of slowish. It takes a second, or it takes half a second for each step. And we seem to need many steps because we haven't got very far yet. Luckily, I moved to our computer with a GPU, where Carsten Rippes group have spent 2,000 euros on buying a GPU, a modern one. So I can just tell it to switch the whole thing to the GPU. In Torch, the GPU is called CUDA, because CUDA is a programming standard developed by NVIDIA, which specifies how software should talk to an NVIDIA GPU. And by simply saying here, taking here, you see here by my starting point, I say take this matrix, which is a NumPy matrix, and turn it into a Torch tensor here. And now I say, and take this Torch tensor and move it to the memory of a graphics card. And for the encoder and decoder, make these random numbers already on the graphics card. Again, as before, I have to tell it that I need a gradient for these, as I did before, because I want to improve them and want to let them optimize them. For this thing, I don't need a gradient because this is just a constant. It's our data. The data doesn't change. Now I use here my optimizer, and now I can run it and let it run much faster. Because now, you see, I make a print only every 300 steps, because if I do a print as before for every step, so just for comparison, this is with the CPU, 1, 2, 3, 4, 5, and now the same with the GPU. So, hence I put here only if the loop index is divisible by 300, write something. What I also did is when I let it run a while and notice that this learning rate is much too fast, it goes down, but after 30 steps it runs out. So I put the learning rate down to 0.003, and then it goes down here and on. And let me quickly see where we end up with that by checking here exactly. I ran it till 7,800 and we got to 0.81, which is not that far from the 0.62 that we got with the PCA. But still, the convergence is a bit slow, and you might argue, what's the point of doing it? We won't get better than the PCA anyway, because we only allow for a linear transformation, and we've already proven that the best possible linear combination is the one that the PCA gives us. So, what we really want to do is to replace this with a nonlinear one. And this is where we now switch to MLPs, to multi-layer perceptrons. Anybody has already heard about them? No? So there's two ways how I can explain them to you. One is very abstract, and the other is a bit clear. And the other is explaining the history behind it. I will start with simply giving the definition, and then I tell you why people came up with this. So I will define an affine transformation. That's a map from some r to v n to some r to vm, such that a vector x is mapped to ax plus b. So this map is parametrized by, so let's call this affine transformation, I call it d phi. And phi is a parameter vector, which consists of all the elements of A and all the elements of B. So you see what this is? It's simply a linear transformation, and afterwards I shift everything. That's what you call an affine transformation. You linearly turn it and do it somehow, and then you add a B. That's, without the B, it would be a linear transformation. Now it's an affine one. And the set of all affine transformation from n to m can, of course, be specified by taking all n by m matrices and all m-dimensional vectors. And if I use one matrix and one vector, I describe what affine transformation I have. And now I simply imagine that I take all these elements as one long vector, which I call theta, and call the parameter vector for this thing. The reason why I imagined is as long vector is because I will give all these parameters to my optimizer, and the optimizer doesn't care whether these things are arranged in a matrix or arranged in a vector, in whatever. It will just calculate the gradient with respect to each of them and then make a step there. This is an affine transformation. The other ingredient that I need is called the ReLU, the rectifying linear unit, or ReLU, which is a fancy way of saying f of, how do I call it? I call it A of x is x for x greater than 0 and 0 for x smaller than 0. So this is simply this function. For negative values, it gives 0. For positive values, it's the identity. This we call the ReLU. And now I make the following thing. I decide I make a chain of things where I come in with my 4,000 dimensional thing. And then I go to R100, and then I go to R50, and then I go to R20. And each of these arrows is always each arrow is a affine treffer followed by ReLU. So I go in with my data vector, I do an affine transformation, and then I get a new data vector, which is now only 100 dimensional. When I set all the negative values to 0, then I put this new vector into the next affine transformation, set all negative values to 0, and put them into a third affine transformation. And then finally I get to 20 dimensional. And this, of course, I can imagine as one big function which maps from the 4,000 dimension to the 20 dimensional. And this, the space of all the possible functions that I can describe in this scheme is described by the 3 matrices in 3 vectors, which sit in the 3 affine transformations. So from the point of view of my computer, I can easily describe it. I use here to make it more easy, I use here the sequential thing, which is just a way of telling Torch, I will now give you a list of functions, and please arrange these functions to be all concatenated, in the sense that if I later ask you what's the function value of the encoder, then take the vector, push it through the first affine transformation, then through the ReLU, then through the next affine, and so on. So here, our Torch calls this a linear one, but it's an affine transformation. It's a misnomer here because it has this vector B. And you can imagine that this is a rather large function class. And depending on how you look at it, you might say, yeah, of course, maybe we get a much better encoder that way than our PCA encoder, with a much better reconstruction error because we have so much more parameters. Or you might say, well, actually, we have just changed a couple of affine transformations. And if you concatenate 2 affine transformations, then the result will always again be an affine transformation. If I do something like A1x plus B, and that's A2x plus B2 plus B1. So you see x here goes through one affine transformation and then through the other. And if I multiply this out, I will notice it's still an affine transformation. So if there weren't these ReLUs in between, our parameters, our function space wouldn't even be larger as before. The function space of all affine transformations which go from 4,000 to 20 dimension doesn't get larger if we split up this affine transformation in 3 individual steps going through 150. But now, because I added these ReLUs in between, I can no longer contract this here to a single function because it's no longer linear. Question is, this function looks so terribly primitive. Can this terribly primitive function, of course, technically it's not true. It's true that it's then no longer the same function space, but is the new function space really that much larger than the previous one, that going through these intermediate steps gives us much more functions than if we would go this directly? And the answer is, yes, it is, and it's very surprising. And it took AI researchers about 40 years to figure out that the simpler is better. They came up with all kinds of complicated functions to put between the linear things to break the linearity. And it didn't quite work, and it started working only after they used this most primitive thing. I will tell you more about the story behind this next week, but now I will just quickly show you how it looks like. So here, see how this is now my improved autoencoder. Before, my encoder was simply here pushing this thing through a matrix. Now my encoder here is, it takes the data vector, pushes it through this affine transformation, through Verelo, through another affine transformation, through Verelo, and in the end I get a 20-dimensional vector out. And for the decoder, it goes the same backwards. One affine from 20 to 50, Relo from 50 to 100, Relo from 100 to the 4000. And then I check here my loss and check how far I get with that. And if I run this now, remember here we got, after 300 we were down at 3,900, and after 8,000 we finally got somewhere close to the PCA. This time I'm already better than the PCA after only 100 steps. And if I let this now run a few thousand steps, then it gets better and better and better. But to really get a good thing, it takes a while, so I don't let this run now. I quickly show you here the result, what I got. You see, I let it run here till 8,000, and then I reached 0.53, which is a good deal better than the 0.62 that the thing before gave us. But the difference is still not that great. See, this was the PCA reconstruction, and this is now the reconstruction with a 3-level autoencoder. So there's still a step missing. Now we're getting to the end of today's lecture, so the missing step will come next week. The missing step will be that our loss function is stupid. You remember the loss function which we did was the mean squared error. But actually, our data is Poisson distributed, and we should take into account this Poisson distribution. And this is a phenomenon we've already seen before. 25. If we go here, where last time when we did this smoothing, you might remember that, where did I do this? No, not here. Is this the right thing? Yep, exactly. Where is this it mutate if else? No, I think I put it in this moving one here. Where we saw that if we use the Poisson thing correctly, we could deal much better with the zeros. You remember that we saw that if we just try to reconstruct, to smooth our data, we always run into this boundary which we get from our pseudo count, and that messes everything up. And the same problem we have here, what we actually wanted is that here where we have the salt and pepper shape, that it somehow realizes this is maybe only every 10th cell has the thing. But it can't see every 10th cell because we have this socket of saying the expression is always at least 10 to the minus 4 because that's what we added. And once we solve this thing, by changing our loss function, by not comparing with the original, not comparing with the log normalized data, but comparing with the original count data, things will suddenly work much better. And only if you put these two steps together, the nonlinearity and the Poisson loss, then you get this really nice result, which then really allows you to see, to get a much clearer picture of how strong the expression really is. And this is a very general thing, which not only applies to the single cell data, but this idea that if you have a right loss function, you can denoise your data with an autoencoder and you reduce measurement noise. This is something which is used in very many applications of neural networks and AI, that finding the proper loss allows you to separate the signal from the noise. But I want to stop here, and next time we will have a closer look at this multi-layer thing, And I will tell you the story how people got to Berelo and how people got it all to this idea of using the multi-thing, which is, as you might remember, last year, Geoffrey Hinton got the Nobel Prize for Physics, even though he didn't actually work in physics, he worked in artificial networks, and the thing he got his Nobel Prize for is this thing. Nowadays it looks trivial, but when we had this idea in the 80s, it really changed everything and brought us, it ended what we call the AI winter. It means people got got a new impetus to try artificial intelligence research again after everybody had given up on it for 20 years. And yep. And this was sort of a starting point, which ended us now in the mess with ChatGPT. Good, rest of the story next week, or the full story. Already got ChatGPT to tell me the story again, in case I remember it wrongly, and maybe I'll also check in Wikipedia. Otherwise, tomorrow I want to try a bit doing something like this. I mean, this was this little homework I put online. I wanted to do this already on Wednesday. I think I only put it on Friday, but maybe some of you had a look. If you hadn't, then we just discuss about it tomorrow. Otherwise, rest next week. And I've just checked 6 people. There's this Eversys lecture evaluation. I think a few of you have already clicked it. There seems to be 6 evaluations, and for whatever reasons they made 2 separate surveys, one for the lecture and one for the exercises. So it would be nice if you fill it out. I think you should have gotten some personalized link by email. Okay. Then see you tomorrow next week.